4 billion trees. Gone. Not logged, not burned, erased by something invisible, imported, and unstoppable. The American chestnut once covered 25% of the entire eastern forest canopy, stretching from Maine to Georgia. It was called the redwood of the East. Then, within 50 years, it vanished. Why did every attempt to save it fail until now? It was 1800, and the American chestnut was the undisputed king of the eastern forest, towering over millions of acres from the Appalachian ridges to the Atlantic coast. Castanea dentata grew fast, sometimes 4 feet in a single season. Its timber was straight-grained, rot-resistant, and prized for furniture, fencing, and railroad ties that lasted decades without treatment. But the tree fed more than sawmills. Every autumn, it dropped an enormous mast crop, protein-rich nuts that sustained black bears, white-tailed deer, wild turkeys, and vast flocks of passenger pigeons. Read that again. An entire food web synchronized to a single tree's harvest cycle. Appalachian tanneries extracted tannin from chestnut bark in industrial quantities. Rural families harvested the nuts and sold them by the bushel. Chestnut was called the poor man's tree, not as an insult, but as a measure of how reliably it sustained people who had little else. What comes next is the part that still doesn't make sense. In decades, scientists would understand exactly what this meant. Abundance is not immunity. For a while, things actually seemed fine. The chestnuts bloomed every spring, dropped their mast every fall, and the forest hummed along as it always had. That didn't last. In the summer of 1904, a mycologist named Herman Merkel was making his rounds at the Bronx Zoo in New York City. The zoo had planted its grounds with native trees, including a stand of American chestnuts that had been thriving for years. That summer, Merkel noticed something wrong. The bark on several trees was cracking and discoloring in a way he had never seen before. Cankers, sunken orange-brown lesions, were spreading across the trunks. Branches above the wounds were dying. The trees looked, from a distance, like they were simply stressed. Up close, they looked like they were being consumed from the inside. Merkel collected samples and brought them to William Merle, a botanist at the New York Botanical Garden. Merle examined the tissue under a microscope and identified the culprit, a fungus that would eventually be classified as Cryphonectria parasitica. The organism was almost certainly imported on Asian nursery stock, Japanese chestnut trees brought into the country in the late 1800s. Those trees carried the fungus but showed little sign of disease. They had evolved alongside it for thousands of years. The American chestnut had not. The mechanism was precise and lethal. Cryphonectria parasitica enters the tree through any small wound in the bark, crack from frost, a scratch from a branch, an insect puncture. Once inside, the fungus spreads through the cambium layer, the thin band of living tissue just beneath the bark that carries nutrients and water between roots and crown. As it spreads, it produces oxalic acid, dropping the local pH so severely that the tree's own chemistry begins to fail. The fungus forms a canker, a ring of dead tissue that slowly encircles the trunk. When the canker completes its circuit, the tree is girdled. Everything above the canker dies. The roots, protected underground, survive. The crown does not. The spread rate was staggering. Researchers tracking the blight's advance in the early 1900s estimated it was moving at approximately 50 miles per year. There was no natural barrier that could contain it. The Appalachian ridges, which had slowed other invasive species, offered no resistance. The fungus traveled on wind, on birds, on insects, on the boots of loggers and hikers moving through infected stands. Every chestnut tree in its path was a new host, a new launching point for another wave of spores. Pennsylvania, watching the blight advance from the northeast, passed the first chestnut blight law in 1911. The state mobilized eradication teams with a straightforward mandate: find infected trees, cut them down, burn them before the spores could spread. It was the same logic that had worked against agricultural blights in controlled fields. In an open forest covering millions of acres, it was futile. The teams could not move faster than the fungus. For every infected tree they found and burned, hundreds more had already been silently colonized, their cankers still too small to see from a distance but already spreading. By the time researchers fully understood how the fungus worked, the blight had already crossed into Virginia. The eradication effort was abandoned. There was nothing left to eradicate, only to watch. The chestnut blight did not announce itself. It crept through the most abundant forest tree in the eastern United States and turned it into a ghost before most people realized what was happening. Picture the eastern forest in 1950. The canopy looks, from a distance, like it always did. Oaks, maples, hickories filling in the gaps. But walk through it and you start to see the stumps. Thousands of them. Enormous, moss-covered, some still sending up thin green shoots from root systems that refuse to die. The shoots grow for a few years, reach sapling growing height, and then the blight finds them again. Cankers form, the bark splits, the shoots die back to the ground, and then, the following spring, they try again. Between the first confirmed outbreak in 1904 and approximately 1950, an estimated 3.5 to 4 billion American chestnut trees were killed by Cryphonectria parasitica. That number represents the largest ecological disaster in the recorded history of American forests, larger in scale than any logging operation, any wildfire, any other single event that reshaped the eastern landscape. In terms of sheer biomass lost, it was catastrophic. But here's the detail that everyone missed at the time. The economic collapse happened almost simultaneously with the ecological one. Appalachian communities that had built their livelihoods around chestnut timber and nut harvests lost both within a single generation. The timber industry pivoted to other species, but the transition was painful. Chestnut had been uniquely rot resistant, which made it irreplaceable for fence posts, railroad ties, and mine supports. The tannin industry, which had depended on chestnut bark, collapsed entirely. The Appalachian tanneries that had employed thousands of workers did not survive the transition. Wildlife populations dependent on the chestnut mast faced severe food shortages in the decades following the collapse. The autumn nut drop had been so reliable, so abundant, that entire populations of bears, deer, and turkeys had synchronized their foraging behavior to it. When the mast disappeared, those animals had to adapt, shifting to acorns and other mast species that were less predictable and less nutritious. Passenger pigeons, already under pressure from hunting, lost a critical food source. The species went extinct in 1914. The same decade the blight was spreading fastest. The connection is debated, but the timing is not coincidental. Oaks and red maples moved into the canopy gap left by the chestnuts, and the forest did eventually close over. But it functioned differently. Chestnut mast had been available every year in enormous quantities in a predictable window. Oak mast is cyclical, boom years followed by lean years and less nutritious per unit. The forest that replaced the chestnut forest was quieter, less productive, and less stable for the animals that had depended on it. The clock was already ticking. The chestnut just couldn't hear it yet. The root systems kept trying, kept sending up shoots, kept reaching for a canopy they could never hold. The species entered what biologists call functional extinction, alive in the most technical sense, present in the forest, but unable to reproduce, unable to complete its life cycle, unable to matter in the way it once had. For 30 years after the blight completed its sweep, the American chestnut existed in a kind of scientific limbo. Researchers knew the roots were alive. They knew the blight was still present in the soil and bark of every forest in the chestnut's former range. They knew that any sprout that reached reproductive maturity would be killed before it could set seed. And for 3 decades, no one had a credible plan to change that. And this is the part where things stopped being funny. In 1983, a plant geneticist named Charles Burnham proposed something that sounded almost too straightforward. Burnham founded the American Chestnut Foundation, known as TACF, with a specific genetic strategy in mind. His approach was not to find a cure for the blight. It was to breed a tree that didn't need one. The strategy was called backcross breeding. The Chinese chestnut, Castanea mollissima, had evolved alongside Cryphonectria parasitica for thousands of years and carried strong natural resistance to the fungus. The plan was to cross the American chestnut with the Chinese chestnut, producing a hybrid that carried resistance genes. Then cross that hybrid back to a pure American chestnut, then again and again. Each backcross generation would recover more of the American chestnut's genetics while retaining the Chinese resistance. The target was a tree that was 15 out of 16 parts American chestnut in its genome, 94%, but still carried the Chinese resistance traits. The process was not fast. Each generation required planting seeds, waiting years for the trees to mature, exposing them to the blight to test their resistance, selecting the survivors, and crossing them again. A single backcross cycle could take 5 to 7 years. Burnham knew this when he started. He started anyway. A tree that takes generations to breed demands scientists who are willing to start work they will not live to finish. So yeah, not great as a career incentive, but the foundation found them anyway. By 2023, TACF and its network of state chapters had planted over 30,000 third-generation backcross seedlings across 16 U.S. states, from southern Appalachia to New England. Decades of patient, methodical work measured in growing seasons rather than years. But this wasn't just about the breeding program anymore. A parallel effort was already underway in a university laboratory in Syracuse, New York. And it was about to change the entire conversation. In a laboratory at the State University of New York College of Environmental Science and Forestry, SUNYESF, two scientists named William Powell and Scott Merkel had been working on a different kind of solution for years. Where TACF was working with genetics inherited through breeding, Powell and Merkel were working at the molecular level, searching for a way to neutralize the blight's primary weapon directly. The weapon was oxalic acid. Cryphonectria parasitica produces it as it spreads through the cambium, dropping the local pH and essentially poisoning the tree's own tissue. If you neutralize the acid before it did damage, the fungus would still be present, but the tree would survive. The canker would form, but it would not girdle. The tree would live. Powell and Merkel found their answer in wheat. Wheat naturally produces an enzyme called oxalate oxidase, abbreviated OXO. This enzyme breaks down oxalic acid into carbon dioxide and water, two compounds that are completely harmless to the tree. The mechanism was already present in nature. The challenge was getting it into a chestnut tree. Using established genetic engineering techniques, the team inserted the gene encoding OXO into the American chestnut genome. The insertion involved a single gene. The resulting tree remained, by genomic analysis, 99.999% American chestnut, but its effect on blight resistance was substantial. Field trials showed that trees carrying the OXO gene demonstrated strong tolerance to Cryphonectria parasitica. Cankers formed but did not spread to girdle the tree. The crown survived. The team named their most advanced line Darling 58 after a major donor who had supported the research. The core argument Powell and Merkel made to regulators and to the public was straightforward. This is not modification for modification's sake. The American chestnut is a native tree that belonged in this forest. The blight is an introduced pathogen that removed it. Inserting a single gene from another plant species to neutralize that pathogen is not creating something new. It is restoring something lost. That argument was compelling to many scientists. It was not universally accepted, and the path from the laboratory to the forest was about to become considerably more complicated. Most people never heard about this part. In 2020, SUNYESF and TACF filed a joint petition with three separate federal agencies simultaneously: the USDA Animal and Plant Health Inspection Service, the Environmental Protection Agency, and the Food and Drug Administration. It was the first petition ever filed in the United States for the intentional environmental release of genetically engineered forestry. Nothing like it had been attempted before at this scale, in this context, with this level of ecological implication. The three-agency review was required because Darling 58 occupied an unusual regulatory space. To the USDA, it was a plant, a modified crop organism subject to agricultural biosafety rules. To the EPA, the OXO enzyme it produced was functionally similar to a pesticide mechanism, a biological agent introduced to suppress a pathogen. To the FDA, the chestnut was a food source for wildlife and potentially for humans, which triggered food safety review. Three different agencies, three different regulatory frameworks, three different sets of data requirements, all applied to a single tree. That's the kind of number that makes accountants cry. Three simultaneous federal reviews, each with its own timeline, its own comment periods, its own scientific standards. The petition ran to thousands of pages of supporting data on blight resistance, ecological effects, gene flow, and food safety. In 2023, the FDA issued a preliminary, non-binding assessment of Darling 58. The agency did not reject the petition. It did not approve it. It flagged areas where additional data would be needed before a final determination could be made. The FDA's specific concern centered on three areas: the long-term ecological effects of widespread chestnut reintroduction, the potential for gene flow from transgenic trees to wild relatives, including native chinkapin species, and the safety of the transgenic nut for the full range of wildlife species that would consume it in a restored forest ecosystem. The petition remains under active review. Powell's team at SUNYESF has continued refining newer transgenic lines alongside Darling 58, incorporating incorporating additional data from ongoing field trials. Critics of the transgenic approach argue that the unknowns are too significant to proceed. Supporters argue that the cost of inaction, a continued functional extinction of a keystone species, is already known, already measured, and already devastating. Up to this point, the science had been winning. That was about to flip into a much harder conversation about who decides what belongs in a forest. Two roads are converging on the same destination. The backcross program has trees in the ground across 16 states, but carries slightly diluted genetics. The transgenic approach offers a purer genome and full resistance, but awaits federal approval. TACF now integrates both. Backcross trees can receive the OXO gene, stacking the strength of each method. The root sprouts still emerging from 100-year-old stumps are, in a real sense, the same trees that stood before the blight. Hit subscribe so you see the follow-up when the forest answers back.